a few minutes to change your title.
Thank you. Thank you. So, congratulations to the Department of Computer Science and Automation on this milestone. And it's an honor to be at this workshop in celebrating this milestone. And I've really learned a lot. I've enjoyed all the talks here. This is not actually the title of my talk, but it's just a way for me to kick off discussion. So I study cells. And I like to ask people what would life look like on other planets, just to focus their minds on what makes biology special. Of course, physics and chemistry would be the same anywhere you go. What makes biology interesting on Earth, at least, is there are some organizing principles to biology. It brings together a really important 17th century idea, a really important 19th century idea, and a really important 20th century idea, and that's what makes biology special. And it's plausible. You could argue that these organizing principles would occur, at least 2 out of 3 of them, on life forms, no matter where in the universe they arose. So today I'm going to focus on, you can expand information to include computation if you want. I'm going to expand on using the idea of computation to understand how cells actually function. And it's nice to see lots of young people in the audience, just for you guys. So Freeman Dyson is, one of those clever physicists, and Paul Nurse is one of those clever biologists. And in relatively recent interviews, they've both said, so Dyson, who was one of the inventors of quantum field theory, says a lot of people who are really computer scientists are doing biology. The line between math and biology is not so sharp. The young people are having the same good times now as we had 50 years ago. So the fun is in biology. You should be doing it. Paul Nurse, who's a bench biologist, he's a Nobel Prize winner who discovered how the cell cycle works. His opinion is we need to make biology more abstract. We need to transform molecular interactions, biochemical activities, biophysical mechanisms into logical and informational structures and processes. He thinks that's the real way to think about biology. And I think you should take these ideas quite seriously. So I'm going to give you an example of that, things that line up with that kind of manifesto. So I'm going to be talking about the synthesis of complex biomolecules, like that. I'll explain what these molecules are, and I'll tell you what we've learned about thinking about this using the algorithmic lens. Before we start, so this work was the work of my former PhD student Anjali Jaiman, whose work is now on the archive, and you can read all about the stuff I'm talking about today right there. There's also an IISc connection, so it really got started when Anjali and I started talking with Arnab Bhattacharya, who used to be in this department many years ago, and we spoke for many years before we really understood how to think about this system. The biology part of it I learned from a good friend, a biologist, Ajit Varki at UCSD. These are the other members of my group, and you can reach me by email there. Okay, so the molecules I'm going to talk about are called glycans. They're called glycans because glyco is like sugar. This is the surface of a cell. That's the cell membrane, and that's about 10 or 20 nanometers. There are proteins, there are lipids and all that over there. But every cell on the planet, for several tens of nanometers above the cell membrane, the first thing you hit is not proteins and it's not lipids. This entire forest is made of sugars. It's actually made of trees made of sugar monomers. And some of you are familiar with some of these things. For example, your blood group antigens, when you say A or B or whatever, those things, those molecules on the blood cell surface are actually these little objects which are trees made of sugar monomers. And those are the things I'm going to talk to you about today. I'm going to spend the first maybe 10 minutes giving you a lowdown on the biology. There is some detail here. You don't have to follow the whole thing, but it's nice for you to know that I'm actually talking about a real system. So the surface of a cell again, but now drawn in a cartoon form. So there are these proteins, which are these sort of yellow chunks over there, but these proteins are attached to these sugars. So the sugars are attached to the proteins. And it's the sugars that convey information to another cell that's testing what kind of cell this is. So the other cell will have counter proteins that will try and match in a lock and key form to these sugar structures and check who it is, right? So this is how cell identity is conveyed. For example, this is how things that want to attack us know what kinds of cells they want to attack. So our pathogens use the sugars against us. This is how, for example, if you take a sponge, we were talking about this over lunch, you can take not a kitchen sponge, but a living sponge. You can break it up into single cells and they come back together. You can break up sponge from 2 species, mix them all up, and they separate out and they go correctly into species A and species B. And the way they know what species they are is because they have these correct sugars in the surface. And this is also very interesting. The reason a rat sperm will not fertilize a mouse egg is because the mouse egg has the wrong sugars. So it's also a species marker. So there's information everywhere. So let me dive into detail here. So this is a protein, and a protein is typically 5 or 10 nanometers in size. And as I said, the protein is covalently attached to these tree-like structures. This is a sort of cartoon of what it might look like. This is a more abstract representation of the same thing. So this bottom end of all these trees that I show will be attached to the protein covalently, and each of these building blocks are literally sugar monomers. You can just think of them as building blocks. Each building block can be attached to multiple other building blocks in a tree-like form. These little numbers 3, 6, 1 and so on, they represent which carbon of the sugar is attached to where, because the sugar has many carbons. Now, the reason sugars are more complicated than proteins is the following thing. DNA or RNA or proteins, they're all chains, and the reason is because a nucleotide can attach to 2 other nucleotides, amino acid can attach to 2 other amino acids. A sugar can attach to 3, sometimes even 4 other sugars, right? And this is what allows them to make trees, and this is what makes the combinatorial space of trees made of sugars so complicated, so large, and that's why maybe cells pack so much information into these structures.
Why is it always trees?
I'll tell you why they're always trees. It's a good question. You might think they might come back together later, right? Excellent question. By the way, so again, in terms of information, you might have heard of, well, anyway, so I'll tell you why they're trees. Now, a little more biology background. If I take a protein and I get it into my lab, I have always millions of copies of a protein in a tube, right? And I can cut the tree using a chemical, and I can send those trees for analysis in the mass spectrometry machine. And what comes out is a list of trees like this. And the number one thing that you see is at the same position on the same type of protein, there are always different trees. They're related, but different copies of the protein might have different trees. So there is stochasticity. Okay? That's the first level of diversity. The second level of diversity is the 2 different proteins. These mucins are in your lungs. HCG is the pregnancy hormone, both from humans. 2 different proteins in the same species will have different collections of trees. So the cell sort of knows which protein it is and attaches the correct tree or distribution of trees to the right protein. And thirdly, the same protein between 2 different species, this is horse CG, this is human CG, could have different trees on it. So any theory you make has to account for all 3 levels of diversity. At a protein, between different proteins in the same species, and between different species. Lance Armstrong, the way he got caught eventually, he had this huge organization selling him erythropoietin to dope when he was doing the Tour de France. And he was using human erythropoietin protein, so nobody could ever catch him. Then they realized that they were growing it in yeast, which is not human, and it had the wrong sugars. So the reason Lance Armstrong got caught is because he didn't pay attention to the sugars. So information rich. Right? Okay. Oops. Got caught, sorry. Okay. So what's fascinating about these sugars is they're grown in a very important substructure of your cells. That substructure is called the Golgi apparatus. You might have learnt about this in school and forgotten about it ever since, like I did. But it's this beautiful stack, like a stack of plates or a stack of pancakes. The Golgi apparatus is actually a factory. It's a factory assembly line for making these trees. What it does is the factory, let's say, is moving upwards. This is stage 1, stage 2, stage 3. There might be 3, 4 or 5 stages. And the building blocks are fed into stage 1. And there's all kinds of little machines there that will attach the building blocks to the protein and then to each other. And then these growing trees get passed to the next one through these little carriers, and then to the next one and so on. And out of the last one comes the final product. Okay? So this is really how it works inside a cell. One of the reasons why I think theoretical computer science has had so much success is because the objects that one is studying, you can actually build real world objects, namely computers, that at least match fairly well the approximate models one is making in theoretical computer science. In the same way, biology is about artifacts. It's about real cells that have a real function. And in many cases, those real functions conform very closely to the kinds of abstractions we make for making toy models. So when I say it's a factory assembly line, it has 3 stages. It goes from stage 1 to stage 2 to stage 3. This is not an approximation. That's how it is. Okay? And there might be a reason why cells have organized themselves in such a simple manner to get things working with reasonable robustness. Okay, so I'm going to tell you many things about this, but basically I'm going to tell you the process of how these trees get grown, and then I'm going to abstract away all the structure and come up with some more general results. So you can stop me at any time to ask me about the biochemistry. Okay, but now I'm stepping back into slightly more abstract space. Are there any questions so far? Okay, so we're talking about these trees. The building blocks are sugars. How do you build them? Now, biology makes things in many different ways, and the title of my talk has the word algorithm in it, so I'll explain why I bring it up that way. So there are many things in biology that can take an input and give a product. An enzyme, for example, is a type of protein that takes some chemical and makes it into some other chemical, right? So that's a specialized input-output system. There are other kinds of systems in biology that can make many different things. So for example, there's a machine called a ribosome that makes proteins. But the ribosome doesn't always have to make the same type of protein. It looks up which types of protein it makes in the gene, and it uses that sequence to make a protein. That gene is the messenger RNA template. It doesn't matter what that means. But by putting different gene instructions, it takes the same amino acid building blocks and makes different proteins. Right? So here's the template. Here's the building blocks. Here's the output. The machine that does that is the ribosome. So this can do things that this can't. Right? This always makes the same product or nothing. This can make many different products from the same building block, which is interesting. However, most of biological production is not templated. Most of biological production looks like this. There's some sort of black box, there's some sort of rule, you put in building blocks and you get an output. And this box has no copy of the template to make a copy of. Yeah? So it's quite a general principle in biology, and many biologists in fact think of the growth of an organism in exactly that way. You take a bunch of nutrients, you feed it into the egg, which is some sort of black box, and eventually an adult comes out of it, or a baby, and what controls that process is the instructions in the genome. Right? But not in a templated way. You don't make a baby by making a copy of a tiny baby. Right? It's not a homunculus. So for the purposes of this talk, I'm going to use this word algorithm, and my definition is the following. I mean, every physical process, you can think of it as an algorithm at some level, but for me, a non-trivial algorithm is one that can accept many distinct inputs. So that rules this one out, because a particular enzyme can accept only one type of input. That's trivial. It converts each input to a corresponding unique output through a sequence of steps. Could be stochastic, could be deterministic, but the uniqueness of the output is the important thing. Okay? Different inputs could be converted to the same output, right? But and a given input is converted to a given output. And my non-trivial idea here is also that it should not rely on a pre-existing template of the output. Right? So in biology, it's pretty clear what that means. Either you have a template or you don't have a template. It's very easy to check. So this is what I mean by algorithm. So what's the big deal about algorithms? Well, a lot of people have been thinking about this, not necessarily in biology, but people have become very good recently at building complex structures using building blocks, okay, which is similar in flavor to what I'm talking about now. So this is I think a real output of something called DNA origami, where you can take little bits of DNA and you can control the stickiness between the two bits of DNA by the sequence, and you just drop them in a tube, heat and cool, and eventually you get this picture. And the picture is of course encoded in which bit sticks to whom. The same ideas have been studied in more abstract models. You can read about more of that in these references. But roughly the idea is, suppose I have a bunch of building blocks which are in some disordered input state, and I have a particular target I want them to assemble into with reasonably high yield, right? And it's stochastic, so there are many ways that this thing can grow into that thing. Some of those ways will take you away from the target, and you don't want that to happen. Some of those ways will get you to the target, but then some things might take the target and grow that also, and you don't want that to happen either. Right? So basically, if you can ensure that all growth paths from some input, some disordered input state lead to the target and not anywhere else, and secondly, you ensure that you can wait long enough so you do hit the target, right? Then your yield will be high. So this is in some sort of biochemical or a chemical version of an algorithm. It says that everything that starts here must get there if you wait long enough and no further, right? Not to many other points. Now, so what I'm saying is there's a connection between yield, which is how much of the input becomes the output. That's a really important thing for a cell, and the idea of the entire process being algorithmic in the sense I described in the previous page. Okay, fine. So with that philosophy, what I want to ask is, based on what we know about how glycan, the sugar tree biosynthesis works, can this process be algorithmic? Based on what we know about how cells work, does it even have a capacity to be algorithmic? If so, then can we use everything we know about algorithms to characterize the structures it can or cannot make? Right? And the answer is actually yes and yes. That's what I'm going to show you, and I'm going to show it to you in sufficient detail so you can see the inner workings of this entire procedure. So in the spirit of all the other talks that I've seen in this meeting, we like to get down and show you why these proofs actually work. Okay, so I'm not just going to give you the results, I'll show you the machinery. Okay, so taking everything that I've read about the cell biology, I've compressed all that onto this single slide. This slide captures all the physics and the chemistry of how this system works. It also answers the question of why it makes trees. Okay, this is how it works. This bottom end of everything is always a protein. Okay, and these trees are always attached to proteins. Of course, I won't show the proteins. On the tree, there'll be a growing tree, and there might be some building block, what I call a monomer, which is going to accept another monomer that's going to be attached to it by some enzyme. Okay, an enzyme is a protein that can come and do this specific attachment. Enzymes are specific in the sense they will only take, let's say, the yellow circle and attach it to the yellow square on the left side, like so. That enzyme will only do this thing. It can't do anything else. Okay? So the thing that's being attached to is called the acceptor. The thing that will be attached to it is called the donor. And the acceptor, importantly, might have branches on it. Of course, this bond is free, but that bond might have, in this case, a simple branch, but it might be a very complicated branch. Okay? So acceptor, branch, this is the whole substrate, that's the donor. Now what does the enzyme do? The enzyme actually can check not just for the color and shape of this, abstractly, but can also check whether it has the right branches. So in this particular case, I've shown that enzyme takes the yellow circle, attaches it to the yellow square on the left side, but only if that blue square is already attached to it. Yeah, so there's a context dependence to the action of the enzyme. So in this sense, you can build up a sort of grammar, which is not point by point, but it's based on what's already happened before, which is very important. I'm going to represent the enzyme itself by this abstract cartoon, which actually just captures what its substrate is and the action that it does. So whenever I show something with an arrow on it, I mean it takes this structure and adds that shape to it in that growth direction. Are there any questions about this? So these little cartoons are actually enzymes, and that enzyme, its action is to take these two things and make that thing. Yeah? Okay. Now where do these enzymes live? They live in these compartments. Yeah, those Golgi compartments that I showed you, those things contain hundreds of enzymes, each one of which has a different property. Each one of which will take a different color, attach it to a different color at a different bond. Yeah, and I know everything about these enzymes. So I'm always going to show the enzymes in this kind of little cartoon over here. And on the right side, I'm going to show the reactions that they catalyze. Yeah, so this is the next level. This is now dynamic. Suppose I pass into the compartment this as an input, or this as an input, or this as an input. What's going to happen to it? You can just look up and say, well, I know what this enzyme does. It's only going to take that input and extend it. It's going to leave those two inputs alone because those two inputs don't have the correct side branch. So you're done. Okay? Any questions about this? So I'll always have enzymes. I'll have inputs. The enzyme will act on the inputs, and there may be many enzymes, as we'll see later, and make outputs. And the outputs will come out of the compartment. Okay? You can ignore this thing for the moment. Here's the interesting thing. Always free monomers are added to trees. The enzymes can never attach two things that are already attached to a tree, which is why you only have trees. You keep on growing, okay? And you never prune. So this is in fact a highly controlled growth system. You're always attaching a correct color to the correct color at the correct branch, and you never prune it down. Nevertheless, there can still be sort of noise in the system. Yeah? And let me explain why this is. All chemical reactions in cells are stochastic. Right? They happen with some rate per unit time, and if you look at a single molecule, it may or may not happen in the time window when you're observing it. Yeah? So if you want to think about these reaction networks, this reaction network is that input structure which could get converted to two different structures. That's how you interpret this, which could get converted to some other structure and so on. You can think of this as a Markov chain with propensities along each of the edges, and so it's a stochastic process. You can go down this way or that way, or you could get stuck and so on. Yeah? Yeah. Well, that's the whole point, right? So that's what I'm going to show you. So you build on the whole tree. No, so you always add just one free monomer, and then you add another free monomer. You never attach two bonds that are already on a tree.
So my question is, does the addition of that induce?
No, never does, because you need enzymes for that to happen. Yeah, good question. You mean spontaneously, by accident? Yeah, no, never happens. Okay, so look at this little picture here. Here, the enzyme that's in the compartment is a guy that adds a yellow circle to a blue square, and this thing has two blue squares. Right now, that enzyme doesn't know where to act. So there's some stochasticity. It might act on the left first or on the right first. If it acts on the left first, it might act on the right, and so on. But remember, the reaction compartment has a finite residence time. If the growing structure gets pulled out of the oven too early, right, it may be incomplete. Right? This is like the Jagannath Puri legend where the guy opened the door and the sculptures were not finished. Right? You shouldn't open the door too early, because if you open the door too early, these are stochastic reactions. It may be incomplete. Your best bet is to put the enzymes, put the input, and wait an arbitrarily long time. Many times longer than the rough average rate of these reactions, then you'll get a complete output, right? You notice the difference between these two. All these structures are indeed going to this terminal state, which I've indicated with a notch. They just didn't get there. If you waited long enough, they would all get there. So you think that's all it takes to make the system reasonably well behaved. You just wait long enough. But you could be in trouble. In this case, here's a compartment where there's a yellow circle to a blue square enzyme, just like that. But I also have a blue square to a yellow circle enzyme. This is bad news, because these two enzymes can drive runaway reactions. They can just keep on adding to each other. And so the longer you wait, in fact, the worse the situation gets. So it's not that you can wait arbitrarily long and reduce the diversity of products, which is what you want to do. So how do you reduce the products over here? Well, you have to maybe modify the enzymes so the runaway reaction is not allowed. You require, for example, that this enzyme has a left branch, and unless that left branch is there, nothing's going to happen, and the left branch is only in the input. There are many, many ways to do this. And I'll generalize this in future slides. I'm just giving examples. And then there's a final thing, which is actually really fascinating. There could be multiple enzymes that can attack the same input to give distinct outputs. It's very similar to this, okay? Except that here, if the enzyme acts on this side, the same enzyme could act on that side and therefore bring you back. Here, I've written it in such a way that if this enzyme acts first, that enzyme can act afterwards. But if this enzyme acts first and there's a blue square on the left, that enzyme requires an empty substrate in order to act. That's what that little diagram means. And so it cannot act. So this is a case where no matter if it's early time or late time, it's just a competition of two different ways that system could go. The system can actually branch, and once you branch, it can never come back together. And this leads to diverse outputs no matter how long you wait. Yeah? And the only way to fix this is to really remove some enzymes from the compartment, which we will make use of shortly. So in all these cases, I'm going to call any compartment that gives many structures with reasonable abundance non-algorithmic, and any compartment that gives a single structure with reasonably high abundance algorithmic, okay, for each input. So here's the more abstract way of saying things. Remember I said if a compartment contains let's say a green to a red adder and a red to a green adder, then you'll get a runaway reaction, right? So these are called runaway reactions. They make arbitrarily long polymers. We can capture the propensity of a compartment to do this without having to know everything about all the enzymes in the compartment. You can reduce it to a very simple monomer to monomer graph, which just says of all the enzymes, if anywhere in the compartment there is an enzyme that can add this to that or that to this or this to that, it's a sort of projection down of the whole set of enzymes. If that graph, I can describe it offline, if that graph happens to have a cycle, then the compartment will have a runaway reaction. Right? This is an if and only if condition. I'm just saying it like this because it's very easy to verify whether a compartment can have a runaway cycle or not. Okay? The other thing you'd like to verify is whether a compartment has these divergent reaction potential, okay, where the action of one enzyme prevents the action of another enzyme. That's also reasonably easy to check. I'll show it to you in this case. In this case, enzyme E1 can act on this substrate, but once it acts, E2 cannot act, because once the blue guy is attached, the green guy is not allowed to act, okay, for some reason. Or symmetrically, if E2 acts first, E1 cannot act. Right? These are just enzymes that are mutually exclusive. One causes the substrate to be modified, and therefore the other one no longer recognizes it as a substrate. Yeah? So this is called an acceptor conflict. Both the enzymes have the same acceptor. They both attack the acceptor. Once one attacks, the other cannot attack. Whichever one attacks first, you're in trouble. More subtly, here's a case where, what's this guy on this side? This is an enzyme which can add a blue to a substrate as long as it only has a green on that side, right? So if it has a green on that side, it adds a blue, no problem. But there's another enzyme lurking around which is going to modify that branch itself, right? No matter how deep that branch is. And far away that branch gets modified with this orange circle. Okay, if that happens first, this enzyme can no longer act. So what's happening in this case is this enzyme has a window of opportunity to act when the substrate is unmodified. But if somewhere else on the structure it gets modified, it's missed the boat, it can't act anymore. And this is called a type 2 acceptor conflict. Okay. I'm just giving you details, but the point is that both these things lead to non-algorithmic output. This one leads to unlimited random polymers. This one leads to divergent reactions. And what we'd like to do is eliminate both of these. Right? And it turns out we can. And it's necessary and sufficient to have algorithmic reactions to eliminate both of these things. Okay? And you eliminate both of these things by removing certain enzymes from the compartments. And it's actually fairly easy to check which enzymes you have to remove. The problem with this one is if this enzyme acts first, that enzyme can never act. So the guys that take this path go down one path. The guys that take that path go down another path. They never come back together. Yeah? That's the problem. If these were very distant parts of the tree, if this one acted first, no problem, that one can act later, or the other way around, you would still converge. This prevents reconvergence. So we call it divergent. Okay? So it turns out once you remove these little cycles, then you remove runaway reactions. And if you remove acceptor conflicts, you remove these divergent reactions. Having done both those things, it turns out that the only way the tree can grow, there are still many ways for a tree to grow because it's still stochastic, but all the growth paths have certain very reliable properties, which are the following. Okay? This one is fairly easy to see. If I follow from a root to a tip of any chain on a tree, if there had been a cycle, the same color would have occurred twice. Yeah? And that could then occur infinitely many times. Instead, what you have is no cycles, which means that any root to tip path along a tree has a partial order of colors. There is a partial order. Yeah? This is just a side effect of having no runaway reactions. More interestingly is on this side. When you eliminate these kinds of conflicts, it turns out what you're actually doing is ensuring that any given substrate will always open up its branches in a fixed order. Number 3 first, then number 2, then number 1, or something like that. The same substrate occurring no matter where it occurs on the tree, once you've eliminated this, the branches always open in the same order. For that piece, not in time, right? The time ordering of growth could be random and asynchronous all over the tree. But at any given place, it unfurls in a fixed order. Once you eliminate these, all growth is depth first, because you always finish a branch before the next one acts. You never want to act on an unfinished branch. Okay? So these are the conditions: partial order along a chain, strict order in the way you fan out the branches, and depth first growth. Okay? In fact, for a tree that starts from a single root, these two conditions simply mean the tree can only grow in a fixed order. The reason I'm saying it in a slightly more complex way, the tree you inherit as the input could have many copies of the same substrate, which could grow in asynchronous ways over time, right? So it's not as simple as a uniform order across the whole tree. Okay? So all this said and done, all it comes down to is the following thing. There are types of variations of molecules, right? You can get the incomplete oligomer because you pulled it out of the oven too early. You can get these random repeats. You can get competing fates. These are properties of the trees. These properties of the trees are equivalent to the properties of the reaction network. Incomplete means you exit early. Tandem repeat means there's a runaway reaction. Competing fate means a divergent reaction. This is for trees. This is for the reaction network. We now have necessary and sufficient conditions. You can ignore this rightward arrow. Anyway, so this is a necessary condition, but anyway, by looking at what enzymes are in the compartment, you can tell which of these things is going to happen. And therefore, I can remove the enzymes and prevent these things from happening. So roughly, it works like the following. If you gave me a compartment with a bunch of enzymes, you just picked all the enzymes in some yeast or something and you put it in there, and then I gave it an input, all the bad stuff would happen. You'll get a runaway reaction. You'll get a divergent reaction where the left and the right could form independently, right? And you could stop, at an incomplete state. All the bad stuff happens. First, you want to remove the cycles. So here's a cycle, for example. I've now broken the cycle using a clever enzyme. Once I've broken the cycle, I know that I can't get infinitely sized trees or arbitrarily long trees. All inputs will lead to finite outputs, but the same input could lead to multiple outputs. Here's a case where if the right enzyme acts first, the left one can't act. If the left one acts first, the right one can act, so you get both these guys. And you get the same things all over the place. Yeah? So this is okay. It's not runaway, but it's still divergent. To remove divergence, I have to remove these conflicts. Right? Once I've removed the conflicts, every input to this compartment goes to a unique output, right? Which is why I call it algorithmic. Yeah? And most importantly, inside the compartment, if I could watch the trees growing one at a time, they would all grow in an order which is called uniform depth first growth. Uniform means identical substrates at any point in the tree grow exactly the same way with a branch order which is fixed, and depth first means depth first in the obvious way. Okay? So what we really, our big result is that algorithmic is something which is fairly intuitive. We've connected algorithmic to the idea of uniform depth first growth inside the compartment. Okay? So this sets us up perfectly to solve the inverse problem, which is actually the thing everybody's interested in. Yeah? Suppose I'm given an input. In this case, the input, so here's a tree, right? This is abstract. This is not what the trees really look like inside a cell. This tree is actually much more complicated than you will find in real cells, but I've built it as an illustration of our method. Okay? So the thick lines and the filled circles, those are all the given structure. Okay? The thin lines and the empty circles are all the new parts that you want to add to it. What you want to do is to dump this guy into the compartment, and you want it to grow to exactly that size and stop at infinite time. Yeah. So that's the input. This is the added bit. This whole thing is the target, and you want that to be the terminal target, which is why I've labeled it with this notch, just like I did in the first slide. Basically, at infinite time, this should be the target that comes out. Yeah. So here's the theorem. An input target oligomer pair, like so, is algorithmically achievable, means it can become the unique infinite time output of a compartment by putting the right enzymes in a single compartment, if and only if there is a uniform depth first growth order from the input to the target. It's that simple. So to check this fairly complicated biological requirement, I want to make this structure. To check whether it's possible, I merely have to take the input tree and run it all the way to the target, and all the different ways I can imagine adding monomers to it. So this is fairly big, right? There are many ways this tree could grow from the input to the target. Walk through all those ways, and among all of them, just find one way which happens to have this depth first uniform growth property, which is something that can be written independent of everything else. It's just dependent on the tree structure itself. Okay? So this is very strong. We'll make it stronger in just a second. Now you might think this is going to be an explosive thing, very, very large in the size of the tree. First of all, the trees are not very large, so I'm not really worried about it. But there's a very efficient algorithm to even do this. To check whether a given input to output pair has a uniform depth first growth order is actually fairly easy. And that works the following way. So what I do is I take the given input tree, and I take all the parts of the tree that need to grow further, right? Because that one is already grown, so I don't care. That one is not grown. That one is, all the others are not grown. Yeah? And I write them all down. This is not a complete set of things, right? This is just a few of the things that I want to grow. I notice that all the orange guys should not grow at all, right? I notice that all the green guys that are empty should grow to this state. I notice that all the blue guys that are empty means initially not attached to anything, should grow to this state, and so on. Yeah? So I write them all down. Now this is actually very interesting. So it turns out that the branches initially can lead to different final states. An empty red, which I call A, goes to this structure, because that's what we've required over there. Yeah? But an A that already had a green attached to it goes to this structure, because that's what I've required over there. So this is my wish list, right? That's my list of things that I want. The real question is, are all these compatible with each other? One simple way they might be incompatible is a red guy on this side and a red guy on that side both have different fates, right? It's a fairly obvious way in which it can be incompatible. This one is incompatible in a very subtle way, and I'll explain. So this, you get tired of making these graphical diagrams, so I'm going to represent all my desired input outputs in a more sort of alphanumeric format. Right? Now when I say that C is the empty guy, you notice that C can sometimes be attached to one other thing, right? So C can have one bond or can be attached to two other things. It can have two bonds. So C has two empty bonds. A C with two empty bonds is just the bare C. A C with a D on the left is I guess that guy, right? A bare C becomes a C with a D on the right. A C with a D on the left becomes a C with a D and a full A on the right, A hat. Yeah? So this is my full list. So for example, I can then represent that on a little graph like so. These are all the movements I need to achieve in order to make this tree work. Okay? If there are questions about this, I can address it offline. So now I write all of it down and I say, can it be done? And in this particular tree, I've put enough conflicts that it turns out I can't do it. I can easily make sure that empty C goes to this structure and a C with a D on the left goes to this structure. That's those two conditions. That's totally fine. B, for example, I can easily make the empty B go to the B with the 2D structure, or I can make the B with the 1D structure also go there, no problem. But with the A, you end up having a problem, because once you write down all these, right, each one has only three ways it can grow, either one or the other, or the third bond has to be attached because it's uniform growth, right? But once you write everything you require down, there is no path that takes you from the top to here. Or if you had a path from the top to here, all the other paths would be cut off. Okay? So this tree contains just enough conflicts that it cannot be made in a single compartment, and it's very easy to verify. So all this is just to show that I have a simple algorithm to check whether this is even possible, which makes the theorem actually useful. Okay. Going forward, remember I told you that actually in a cell you have many compartments. What's the point of having many compartments? Other things you can make in many compartments you can't make in a single compartment. So what happens is you grow a bunch of things in first compartment, move that growing tree to the second one, where now the rules are different because there are different enzymes attaching different things to different things. Right? So in general, you start with an input and it goes from one compartment, spends some time, to another compartment, spends some time, and so on. It's quite complicated. And this could exit in an incomplete fashion. It could exit after divergence. I mean, all kinds of stuff could happen. Right? But it turns out it's tame in the following sense. If I want to again check, can this be made into that? Here's the theorem. An input-target oligomer pair is algorithmically achievable, unique output of the system, in n compartments. Okay? If and only if there is a growth order from the input to the target that can be fully decomposed into these kinds of growth orders, n uniform depth first stretches. Okay? So this is very easy to check again. In fact, if you don't care what the value of n is, you can actually check whether something can be made by a series of compartments, each of which contains a single enzyme. Those are very easy to understand. So that's a very simple corollary. Input-target pair is algorithmically achievable if and only if there is a series of single enzyme compartments that converts the input to the target as the unique final output. And it turns out this is just trivial to check. You can do it on a napkin. Yeah? So let me put this to use. Here's a very surprising example of a structure which is algorithmically unachievable. It cannot be made as the unique final structure in any number of compartments. Yeah? And the reason is because it has just enough competing guys. You know, if an enzyme extends this blue, it should also extend that blue. If it extends this yellow, it should also extend that yellow. There's no way that an enzyme can be told just do this and stop, or just do that and stop. So this is not achievable. Very easy to check using the theorem I just said. Here's another one that is trivially not achievable. If I have a system where I want the yellow square to end up with 2 yellow circles, that enzyme is going to keep on making more yellow circles. It just cannot stop. That's not achievable. Here's a system which I can apply this kind of calculation to and check whether there is a path, and it turns out it is achievable. Then I can go back and check how many n independent uniform depth first growth stretches. It turns out it's 3. So this can be achieved, but not in one compartment, not in 2 compartments. It needs at least 3. So this is a very biologically relevant way to characterize the complexity of a structure. How many compartments do you need to make it? And I can actually go inside cells and count how many compartments it has by seeing what structures it makes. Okay. So I'm running out of time. So what I'll do is I'll skip this sort of many benefits of compartments and get to the real data. This is real data. Mucins are these things in your lungs that protect you from the outside world. These are mucins from a cystic fibrosis patient, not our experiments, they come from online data sources. Okay? This is human CG, HCG, which is the growth pregnancy hormone. This is the horse version of the CG, and these are the trees attached to those. This is all real data. Yeah? So what we can do is infer the smallest set, this is not unique, but the smallest set of compartments that can make these starred structures and not others. Yeah? And it turns out that in these 2 cases, you need 3 compartments. In the last case, you need just 2. The first, second, and third compartments contain these enzymes. Because they contain these enzymes, the compartments do these things. And if you stare at it a while, you can realize why this reaction happens in that compartment. Okay. What's interesting to me is the following thing. These 2 collections of trees look very different. But in fact, the main difference is that a certain enzyme moved from the last compartment to the first compartment. Some instruction got done first instead of getting done last. And that changes the whole thing. Yeah? So small changes in the code lead to large changes in the output, which is fairly obvious, but it's nice to see it happen in real data. This one is even more interesting. What happens in the horse case, it looks very much like these last 2 compartments have just fused into one. And whereas previously there were no cycles, now there's a cycle, a yellow to blue, a blue to yellow. And that's why you get these long yellow blue type of things in the horse case. Right? So again, a very small change has led to a very big change in the output. Now this is all very detailed, but a nice way I like to capture this is I like to imagine the following thing. Imagine that I've done all these reactions and I'm just examining what's happening in the final reaction. Imagine that I'm able to look in and see exactly what structures are there, get the whole distribution, everything. And I wait as a function of compartment residence time, and I pull it out and I plot the Shannon entropy of the collection of structures that you get out. Yeah? The entropy starts at a certain input entropy. That's what this is. That's what you inherited previously. In all cases, the entropy goes up initially. That's because you get all these incomplete structures. but eventually you complete them, right? So the entropy goes down again, unless you exit early, which is what's happening in this mucin case, or you wait too long and it's a runaway reaction, in which case the entropy doesn't go down, right? So I think it's a very nice way to control the diversity of systems. Either you wait long enough, right? Not either. First of all, you remove the runaway reactions, and then you wait long enough for the thing to reach completion. Okay. So this is the message. It's sort of much easier to copy a template than to follow a recipe, and biology mostly does the latter. And I'll take questions. Thanks. Ask me about the biology also. I'm happy to answer. You can ask me over coffee. Okay, good. So catch me offline. And again, thanks for having me at this meeting. It's been really a lot of fun.